In this video, we will be investigating the process of cellular respiration and the various stages, including glycolysis, Krebs cycle, electron transport chain, and lactate fermentation. Let's begin. The first step of cellular respiration occurs in the cell cytoplasm, often in the absence of oxygen. This glucose molecule, a 6-carbon structure, will be phosphorylated by the investment of 1 ATP. Glucose will undergo another energy investment in which its other side of the chain is also phosphorylated, requiring the investment of another ATP. Now we have this 6-carbon structure which is phosphorylated on both sides. This is where this unstable structure will break down into two different molecules known as G3P. Next, this G3P will be phosphorylated, and in the process, an NAD+ is reduced into an NADH. Keep in mind that this is going to happen on both sides, but for the sake of simplicity, we're going to only show one side for now. So in reality, we have the production of 2 NADHs from this entire phase cycle of glycolysis. Next, what's going to happen is this G3P is going to break down. It's going to lose one of its phosphates, and as a result, it's going to release ATP. So now we have the energy payoff phase. And then the same thing is going to happen one last time, creating just this G3P molecule, which is eventually going to be converted into a compound known as pyruvate, which is simply a 3-carbon compound that will enter the next phase of cellular respiration. So out of this entire process, if you were to track down the numbers, you would realize that we had an investment of 2 ATPs, and we had a payoff of 4 ATPs for both molecules of G3P, making our net 2 ATP, and in total we produced 2 NADHs. Keep track of these high energy electrons such as NADH and the number of ATP we are producing throughout the various stages of cellular respiration so we can recognize how much energy we are extracting from this one molecule of glucose. Now we're going to go head on to the next step, which has to deal with this 3-carbon compound known as pyruvate. The next step being pyruvate oxidation. In pyruvate oxidation, this 3-carbon pyruvate molecule is going to be degraded into a 2-carbon compound known as acetyl-CoA. This occurs when 1 carbon from the pyruvate is cleaved off to produce the byproduct CO2. In the process, 1 NAD+ is reduced into NADH. Now, this 2-carbon compound attaches to a coenzyme A to produce the first component of the Krebs cycle, acetyl-CoA. Let's take a look at the Krebs cycle to see exactly how this compound comes into play. This 2-carbon compound, acetyl-CoA, that was produced via pyruvate oxidation, is now going to enter the Krebs cycle, located in the matrix of the mitochondria. This 2-carbon compound starts off by combining with this 4-carbon compound known as oxaloacetate. It ends up forming the infamous 6-carbon compound from which this cycle gets its name, citrate. Through a series of reactions, this citrate is going to be degraded and eventually going to lose 2 carbon dioxides, overall, Krebs cycle produces only 1 ATP, 3 NADHs, the high energy electron carrier we talked about in glycolysis, and a new high energy electron carrier known as FADH2. This occurs for 1 molecule of pyruvate. For 2 molecules of pyruvates, we are going to produce 2 ATPs, 6 NADHs, and 2 FADH2s. Remember to keep this in mind when we do our final count. The electron transport chain is located on the invaginations on the inner membrane of the mitochondria, known as the cristae. This is where various proteins are located that allow us to transport electrons and produce a proton gradient to funnel ATP synthase and produce more ATP. So believe it or not, there is actually a reason why the electron transport chain is referred to as oxidative phosphorylation. For starters, the NADH and FADH2 that we made from glycolysis and the Krebs cycle are going to end up being oxidized. These electrons produced by the oxidation of NADH fall to lower and lower energy levels. Eventually, oxygen comes and picks up the electron and converts it to water. In this instance, the oxygen is being reduced to form the byproduct H2O. Next, the protons also produced by the oxidation of NADH are pumped into the intermembrane space of the mitochondria. From there, those hydrogen ions are funneled through a complex known as ATP synthase, a protein through which the hydrogen ions move from an area of high concentration to low concentration down their concentration gradient and creates a turbine-like movement that sticks an phosphate group onto an ADP, producing ATP. One thing to note is that from each NADH, 3 ATPs can be produced, and from each FADH2, only 2 ATPs can be produced. Assuming that respiration occurred as efficiently as possible, this would produce 38 total ATPs. Now, lastly, we're going to discuss what happens when cellular respiration doesn't occur in the way that it is supposed to, specifically in the absence of oxygen. In the absence of oxygen, anaerobic respiration will occur. One type of anaerobic respiration involves fermentation. This example has to deal with lactic acid fermentation, which occurs in many of our muscle cells whenever we are overly exerting ourselves and our body can't meet up with our energy demands. What happens is this glucose molecule undergoes the same process of glycolysis to produce 2 ATPs and 2 NADHs. In addition, 2 pyruvate molecules are produced. These 2 NADHs that were produced in glycolysis cannot be regenerated back into NAD+, and this poses a problem since we were not able to oxidize that NADH in the electron transport chain. As a result, what happens is this pyruvate molecule actually turns into another molecule known as lactate. This lactate allows for the oxidizing of NADH back into NAD+, thus allowing the cycle to occur once again.